Good morning students, welcome to standard 12 online tutorial. The chapter that we are currently studying is the sexual reproduction in the plants. Now in the last lecture we had talked about the outbreeding devices, we had also talked about the pollination, the types of pollination, okay, that is the self and the cross pollination, and we had also talked about how does the pollination occurs through biotic and the abiotic components. Now today we shall learn about a new topic, that is the pollen-pistil interaction. Now normally what is the phenomena that occurs in the plants is that the anthers, they produce the, we can say, pollen grains. These pollen grains are being transferred to the stigma of the pistil. This is the stamen, filament. So when the pollen grains they transfer to the stigma through self or the cross pollination, this pollen grains they develop the pollen tube, and when they enter into the ovary or the ovule, they enter it through the this is the micropyle end, and this is the which end? So when the pollen grains they transfer onto the stigma, by the help of the pollen tube development, they enter into the ovule through the micropylar end. Now this micropylar end, they have an apparatus known as the filiform apparatus. They have the what children? Filiform apparatus. Now over here, it is interesting to know that not any pollen grains or not every pollen grains they make up to the stigma. Even if they make it, this pollen grain or the stigma will not allow the pollen grain to dehisce into the, we can say, the ovule, as because there are certain restrictions being imposed. Like we can say, not everybody can enter our home. Why? Because we only allow those individuals who we know and we are likely to know, we can say, or those who are relatives. So in that case, the pollen grains also, when they travel onto the stigma does not allow every pollen grain to enter, as because it's a very interactive process that occurs between the pollen and the pistil children. Okay, and due to this interaction, there is an interference of the chemicals also. Now chemical in the sense, if you heard about the structure of the female gametophyte, they have mentioned the term germ pores. Now germ pores will allow the, we can say, the pollen grains to dehisce into the what we can say, the ovary. But as a result, only few of the anthers, we can say, or the pollen grains will be able to make it to the what class? The ovule or the ovary children. Now the next topic that we are going to study is about the artificial hybridization. Now artificial hybridization, many a times farmers they want some ideal characteristics for their growth in their crop, we can say. Now for example, we can take the sugar cane. Sugar cane, it has to be, the stem has to be thick and it has to be juicy. Now both the characters cannot be present in one sugar cane. So what does the farmer do? They take the sugar cane or the genes from the sugar cane which has thick stem, and juicy from genes from the another, we can say, sugar cane. Now what the farmers will do? They will hybridize. Hybridize meaning when two or more desirable characters are being wanted in a single plant. It is being done by mixing that genes with the other genes to make a desirable product children. Now Over here, for artificial hybridization to happen, there are two techniques which are being used. The first one is the emasculation, and the other one, that is the bagging. Okay children, now let us talk about the emasculation first. Now this to both the process, they occurs in the flowers which are having, or the plants which are having bisexual flowers. Please note that the emasculation and bagging occurs in those plants wherein they have the what children? Bisexual flowers. Now over here, this emasculation process means what? See, first of all, when the anthers, we can say, when they are about to mature, the pollen grains, that is this part, they are being plucked out from the flowers before they even mature, or when they are about to mature. This process is known as the emasculation, and the flowers which I am collecting is known as the emasculated flowers. Now these emasculated flowers, they are being protected in a bag-like structure or a suitable cover-like structure, plastic sheet, which is often made up of butter paper or butter plastic bag, we can say. Now why it is being done? It is because to prevent the contamination of the pollen grains, okay, with the stigma. Now that process of covering the anthers or the pollen grains with the covering of plastic bag of suitable size with the butter paper or something, it is known as the bagging, children. It is known as what, children? Bagging. Now the other desirable characteristics of that genes or that pistil is being collected from another species of sugarcane or something, and that is being mixed with the emasculated flowers of that sugarcane or any other plant that which we want to, and after that they are being grown in a suitable condition, okay, that is the in vitro conditions, and after that the desirable characteristics of that plant are being, okay, obtained, children. So this is about the what we can say artificial hybridization and the emasculation and the bagging process. Now the next topic that we are going to study is the double fertilization. Now double fertilization already we have studied in the 11th standard, children, but let us revise. That is, this is the structure of the embryo sac, and these three are what, children? Antipodal cells. This is the polar nuclei. And these are the two synergies, and we can say exam. Now normally the pollen grains, we can say, contains two male gamete, and this is the pollen grain. Now the pollen grain, the one male gamete fuses with the polar nuclei, that is N plus N plus N, will give the what class? Primary endosperm nucleus. That is primary endosperm nucleus. Okay, children? Now, this one will fuse with the egg cell to form the zygote. Now, once the zygote is developing, it requires nourishment. That nourishment is being provided from the primary endosperm nucleus. Okay. Now this primary endosperm nucleus later on develops into endosperm, and the zygote will develop into what class? The embryo. Now in the post fertilization events, children, okay, the ovary develops into fruit and the ovule develops into the seed. Now as over here the fertilization occurs twice, it is known as the double fertilization. Okay children? It is very easy, as we have learned in the 11th standard also children. Now prior to this next video lecture, we will be talking about the structure of the embryo, okay, and the endosperm and the other remaining topics children. So I hope you are clear with the topic of the pollen pistil interaction. How do they occur? Not all the pollen grains, they, what you can say, reach the stigma and develop the pollen tube or they undergo fertilization. Even we had talked about, we can say, the artificial hybridization, which involves two techniques, that is the emasculation and the bagging children. And how does the double fertilization takes place children? Okay children, so this is for all this lecture. Okay, and in the next lecture we shall continue with the structure of the, okay, structure of endosperm and the other structures as well. Thank you.